is a professor at the Department of Zoology at the University of Wisconsin-Madison. And is a member of the board of Beyond Pesticides. So we'll give Warren 20 minutes. That's what we've tried to do with each of them, which will leave us maybe 10 minutes for questions. But I really want you all to have an opportunity to ask some questions and get them answered. Warren?
Thank you, Rout. We've had wonderful talks today, and the talk that I would like to present now is some of a way that we've been working toward to be able to tip the balance so that we have a massive amount of population that wants organic, they demand organic, and they throw out the kind of administration that we have, not only at the federal level, but many state levels too. I want to, let's see, page down, where is that page? There we go. That's still not working. It's working, not working. Oh, maybe I'm doing the wrong one here. There we go. Introducing myself, I'm one of several hundred independent researchers sifting through the peer-reviewed literature and asking, are our data consistent? Do our experiments have good controls? What are the basic principles behind pesticide actions at multiple levels? And those are fats dissolve in fats and opposite charges attract. And what are the literature data? Are they consistent with the data that we're collecting and with theory? I want to show you two graphic slides. The first one is what's happening to human sperm counts globally. As you can see, there's a very substantial decline, about 2% per year to 3% per year. If I go to the next edition here, one of the things you see here is that we've gone from 60 to 70% U.S. males are qualified as sperm donors in the mid-40s to 6 to 7% U.S. males qualifying by 1990, and there's also more recent data for Israel, for example, where only 1% of their soldiers are now qualifying for a sperm donor. And in 2013, we had the very first situation where U.S. whites had more deaths than births. As of 4/26/17, a couple days ago, we had 1 in 8 couples with fertility problems. And a lot of this appears to be being driven by the fact that all of us are conceived as bisexual organisms. Very few people understand that. And we develop our particular sex when we are born, not so much anymore as we heard earlier this morning, at least, but we develop our sex late or partway through our embryonic life. We have two, a male and a female primitive reproductive tract, and at a certain decision point, in parts per trillion to parts per quadrillion, sex hormones of testosterone and estrogen, our fetus, when we were a fetus, decided whether we were going to be male or female. And as you have heard, this is becoming more ambiguous as estrogens or pseudoestrogens in the environment are making our embryos confused as to just which sex they belong to and what their inclinations will be. So not only have we been reducing our capacity to reproduce, but we're also dumbing down our population. As you can see from this CDC graph of 1975, when 1 in 5,000 births was autistic, and in 2014, 1 in 68 births was autistic. The consequences of that for even the population to be able to vote intelligently or make other kinds of critical decisions is in doubt. This is my particular version of how I'm trying to understand the interconnections, or how everything is interconnected, all the way from the mass and energy and nutrients that make up our chemical biological systems, and how that supports individual organ system functions of central nervous system, endocrine, immune systems, and how these molecular and organ system functions support individual functions of reproduction, growth, and behavior, And that supports population level things like birth rates, death rates, and social structures, and community level functions like immigration, emigration, and relative abundance of species. We began to develop this back in 1999 when we first published this paper showing that we could get neurological, endocrine, and immune effects from a single or a collection of herbicides and insecticides and even nitrates. I also want to point out to you that many of these effects, because they're down in low parts per quadrillion to parts per trillion, are inverse dose responses, where the greatest effect is at lower doses. I also want to point out a couple other things, that the gut, as we've heard before, is very important in immune function. But since the immune and the nervous and the endocrine system all talk to each other all the time, it's a coffee clutch of communication with about 50 to 60 molecules. You hit one corner and you hit all three at the same time. And that's one of the things that we were out to show in this paper, and we're able to demonstrate that. I also want to point out something else, and that is that normal development and processes that drive our chemical operations in our bodies, whether we're humans or animals or plants, is driven by the expression of genes. But when you start getting chemicals in the environment, these things are capable of hacking the genetic code and modifying the way that things are functioning, as we just saw illustrated in the autistic and the changes in our fertility. And then there's one more thing that David was, of course, pointing out, and that is the importance of beneficial soil or gut microorganisms, and how Roundup knocks those things out by stopping, among other things, amino acid syntheses, which have antibiotic effects. And of course, Don Huber told us a lot more about many other things that Roundup does. I want to acknowledge, before I go on, a key person who's helped in the development of some of the things I'm going to show you, and that's Dr. Fariba Sadi-Porter from MetResponse LLC, and also in the Department of Zoology at UW-Madison. The Ecotoxicology Research Fund has been a very important part of funding this. The Lumpkin Family Foundation got it started. Mrs. Rodale was a key person in developing it, and then other organizations there, and lately the Sirius Fund that Jim Riddle has been associated with. We've had a list of all kinds of things from Don and others about what are some of the known things that Roundup, which is really a mix, of course, of not only glyphosate, but surfactants and non-ionic solvents can do, and where they are in our diets. And here's just a partial list. DNA damage, which alters gene expression, and that causes changes in developmental patterns. It reduces the enzyme aromatase that converts testosterone to estrogen, which means sexual changes in development and in sexual preferences. Alteration in energy metabolism, like mitochondria, which induces oxidative stress, which kills nerves. It alters immune function. It alters hormone levels, which are the basis for more than 10 different chronic diseases when you start changing things around, especially suppressing immune function. One of the things that hasn't popped up for the most part, but I found this recently, is that Roundup can also stimulate the retinoic acid pathway, which produces then too much vitamin A, and vitamin A is crucial to keep low enough so that you don't induce a lot of birth defects by things which give you things like microcephaly or tiny heads. Of course, it's mostly known because it shuts down the shikimate pathway, which means loss of aromatic amino acids, as Don's talked about, loss of beneficial bacteria in the gut, which David really hit very nicely, and that gut, again, is responsible for about 70% of our immune functions in the gut. And as has also been mentioned, this stuff, because of use as desiccants as well as pesticides, winds up in all the whites of our diets, like sugars and our grains, which means that the contamination is just ubiquitous. And most of us apparently are already excreting glyphosate-rich residues in our urine. And if you'd like a complete summary of this, just Google Pan glyphosate monograph, which came out in 2016, which documents these kinds of things and many more extensively. So I'm going to go back just a little bit to basic principles about pesticides, why entry is easy, and what are these other or inert ingredients and why are they important. If we imagine we've got a cross-section of a leaf right here on the bottom, and we've got this lipid waxy covering on the leaf, we want to get through that. And if you're going to have a pesticide that's going to kill a plant or an insect, you've got to get it inside the organism. So what you do is you add lipid solvents. These are non-ionic or no charges on them. If it's like soap, for example, some of the aspects of soap have no electrostatic charge. Fats will dissolve in fats if they don't have any electrostatic charge. And then the other way to get in is through the stomates or the breathing pores, the spiracles of insects, or in our case of us, our lungs. All of the breathing surfaces are protected by a sort of a hemisphere of water film, and you need to penetrate that, and so you put in surfactants to weaken that surface tension, and that way you get rapid penetration that way. Now, one of the key things about EPA registration process holds, and this was mentioned a little bit by Amy this morning, is that they are not part of, these surfactants and solvents are not part of the EPA registration process, which is crucial because that means that functionally we have a bait and switch registration process. That is, we test just the active ingredient and not look at this. And so that means that we're missing a huge part of the functional aspects of how these mixtures operate. And the consequence of that is, and I'm thinking, have I got that? No, okay. But basically a molecular bull in a china shop. Whoops, didn't mean to do that. How do you design a pesticide to kill? Typically, you will make a ring-shaped structure. Either all corners or all 6 corners of this will be carbon, or in the case here, we've got a nitrogen every other location on this thing. This is sort of the master design for the herbicide atrazine. If you've got a ring-shaped structure like this and it doesn't have any electrostatic charge, that means it's lipid soluble. That means you've got a master entry key for all cells in the body. And then you want to also, though, have some aspects of it being water soluble. So you'll add charged groups. This is a nitrogen with 2 positive hydrogens. The water solubility is there because of the electrostatic charge. So now you need to get it inside the cell. And the way you get it inside the cell is the fat solubility. You dissolve in the fats. Remember the principle, fats dissolve in fats, so that gets you through into the cell. And now there's one more principle, namely opposite charges attract. And so this positive charge here will be attracted to anything that's negatively charged inside the cell. And we've got negative charges on the powerhouse of the cell, the mitochondria, opposite charge of negative charge on the DNA, which means that something like this could go over and slide between the rungs of the DNA ladder, or it could be going up and shutting off the electron transport chains, which is the powerhouse of the cell, and that's how you can kill it. So this kind of a design of both fat solubility and lipid solubility properties of these molecules allow us to have what we call a molecular bull in a china shop. It can interact with almost anything in a cell, and so you can get all kinds of wild results from basically the same molecule, but doing a lot of different things depending on where and when it gets into the cell. Also, I want to point out that a lot of the GMO things that I talk about are two particular kinds of GMOs, not all of them. The GMO crops that are pesticide resistant, like Roundup, which has glyphosate as the active ingredient, and PP GMO crops, pesticide producing ones, like BT corn, Bacillus thuringiensis toxins, which are designed for larval control, but as many people, especially Donna's pointed out this morning, this has a lot of other consequences as well. And then we can have atrazine, which is weed control, and so you can get all of these things in one particular group or agricultural product. The way these BT toxins work, the corn cells produce a prototoxin, and then when it gets into the gut, it changes the prototoxin to an endotoxin. That starts punching holes in the gut lining. The cell begins to rupture, and you have rest in peace. Oops, sorry. We would hypothesize, if we're punching holes in the gut because of the inflammation that Don talked about and David talked about, that we might see things going from the gut into the body. Is there any evidence of that in humans? This particular paper that came out in PLOS One in 2013 deals with that particular issue. There were 4 studies done. Over 1,000 human subjects had their serum analyzed for foreign proteins, especially DNA, and the results were that in the over 1,000 human samples from 4 independent studies, we report evidence that meal-derived DNA fragments, which are large enough to carry complete genes, can avoid degradation and through an unknown mechanism enter the human circulation system. And finally, in one of the blood samples, the relative concentration of plant DNA is higher than the human DNA. This is in a human. So it shows, and the other thing that's unnerving here is that cells, when they get a chance to access DNA, will incorporate it into the cell very rapidly. A way that mating success can be altered is by modifying the relative concentrations of testosterone and estrogen. These 2 hormones come from cholesterol, which goes through a series of reactions here to get to testosterone. And then there's this key enzyme aromatase, which is the bridge, the one-way bridge for testosterone to go to estrogen. If you have atrazine contamination in the body, that will increase the amount of aromatase, and so that makes this whole thing go from left to right faster, and so you elevate the amount of estrogen, as described in this particular paper. If you elevate estrogen in a male embryo, for example, the consequences is lower sperm production when you reach puberty. You may have both ovaries and testes, as Tyrone Hayes has shown in frogs, but it's also, this has been showing up in humans down in South Africa. You might get mammary tissue in male breasts. And finally, how many? We are all done?
You're up.
I'm okay. I'm all done. Okay. How many?
1 or 2 minutes.
Okay. Changes in sexual orientation. So we got soybeans, we got corn, and we have the ability to either hyper-feminize or hyper-masculinize. If we do that for women, that might be behind part of polycystic ovary syndrome. Is there evidence for reproductive impairment? There were 3 papers here showing psychosexual differentiation and diethylstilbestrol in humans, showing that sexual preferences can be altered by pseudoestrogens early in embryonic development. Finally, there are all kinds of neurological things. Here's an example in Mexico from a paper by Elizabeth Gillette. These were normal children up in the mountains of the foothills of a valley in Mexico where they grow winter fruits and vegetables for the United States. These ones here were not exposed to pesticides. These were down in the valley, and these kids had very different kinds of drawings as 4- and 5-year-olds. And so we had now that was back in 1998. Now they've grown up to be sexually mature, and these have very, very tender breasts. The Valley teen girls have only fat in their breasts. They cannot nurse. There's no way they could. And the mothers have very high cancer rates. So our children's future. This is what does this particular picture suggest to you? The genetic control of development has been changed, hasn't it? And finally, a lady who's developed a way to look at this in a very complex way to illustrate how chemicals can be changing the fundamental biochemistry of your body. If you have 10 parts per billion atrazine, there's a whole suite of biomarkers that she can identify. These are the ones when gone down from controls. These others have gone up. There's quite a few different biomarkers that are altered by the exposure to atrazine. If we give them nitrate instead, we're looking at these blue changes. And atrazine and nitrate together, which tend to confound each other, give us only one particular signal. But what it illustrates, and this technology that Dr. Assadi Porter has developed, is a potential way to fingerprint chemical exposures, which could be the data that we need to be able to convince a much wider proportion of our population that GMO is bad. So I'm quitting. Thank you.
So we're supposed to be in 5 minutes at lunch, but I really would like a few minutes for questions. So if all of y'all can come to the front and
Hi, I have a question for Dr. Huber. You mentioned that cancer is peaking in 25 to 40-year-olds. Can you give me where I could find that in some published study or?
But that would be, I can't give you a published study right off. If you look at the CDC data, I'm trying to think. The experimental data would be Seralini's study, 4 months versus 23 months on his study. Cancer started popping up at 4 months versus 23 in the rats. The other one would be the Monsanto study that has just been released, or just what do they call it? Where the court's desealed or unsealed, and that Anthony Samsel will be publishing in the next couple of months. He said it's just about ready. And it'll show that same major shift over to the incidence of cancer on a time basis.
Question for David. You mentioned you visited 6 farms on how they improved the soil. Did you ever visit any where they're applying the biosolids to improve the soil?
I don't write about those in the book, but I have visited farms that have done that, and I do write about biosolids in the book. But of the farms that I write about in the book, I didn't write about a biosolids one, which is a convoluted answer.
Did they improve the soil, the biosolids?
Yeah, the places that I visited that sort of post that, there's been major increases in carbon, and it looks like soil health, but the soil health has not been directly measured. Mostly they were measuring soil carbon. But I think that in the sort of the biosolids world, it really depends a lot on what your source material is, how it's being made, how it's being processed, and how it's being applied. And there's a huge range of practices from what I can tell in that. The place that I went that had the most impressive track record of doing that was the city of Tacoma, just south of Seattle, where they're making TAG Grow or Tacoma Grow. And they've converted their whole municipal sewage treatment plant into essentially a biosolids production facility, or they like to call it a soil factory. and they sell a lot of it back to urban gardeners in town, and a lot of the rest goes to agricultural land in eastern Washington. They put a lot of effort and work into not pre-treating, but getting heavy metals out of their waste stream before they started the biosolids production. So there's all kinds of issues around that, but the short answer would be that yes, the places that I visited that were applying biosolids had seen positive results in terms of soil carbon. There's other places where I've seen the application where I would question whether that was actually working. But
Because we produce 550 tons a day of biosolids, and we give to the farmers and increase their yield per acre, and it keeps the soil more saturated.
yeah,
So that's biosolid. They has improved. Now.
one of the things, you know who one of the earliest proponents of biosolids was? Justus von Liebig, the guy who invented essentially the grandfather of the fertilizer industry. If you read we all know the product of his first book in the 1840s. He wrote another one in the 1860s near the end of his career, where he advocated the large-scale reintroduction of urban sewage after composting back onto farms in Europe as the way to maintain fertility over the long run. So the godfather of the modern fertilizer industry was actually a major biosolids proponent.
Because we find that we treat the poop and the human the personal products, but some of the manures are not treated, and they're using that to improve the soil. That's one thing biosolid has over the manure.
Well, I think that in terms of both biosolids and manure, the composting and treatment is essential to actually making it a good product.
Okay, this question is for Professor Huber. So I have a question like, how do you get rid of the pesticides once it's in your system? Is there a way to clean themselves? Like, what do we do?
Had a little section on that, but I was too long anyway. Start with, you need to know how much you have. We have 3 laboratories now in the States that you can have your urine tested or other tissues tested if you wanted. We've done other tissue testings for infants or for small children who have had extreme pesticide exposure to see where it's accumulating. Glyphosate accumulates in the brain and the bones especially, but in all organs. But you'll excrete a lot of it through the urine, so that most people, to get a good idea of how much you have, will do it through just a simple urine test. Microbenotech laboratories in St. Louis, the HRI laboratory in Iowa, and then Resco Lab in California will all do that. It's about $90 to $100 a test. But give you an idea of whether you need that. Dr. Monica Kruger at Leipzig University has done a lot on how do you remove it. And in Europe, they actually market a product, Activamon, which is about 200 milligrams of humic acid. It's better if you'll mix it with some raw sauerkraut effluent, because that's your biology, or with some clinoptilolite, which is a form of zeolite. That will remove it. If you have infertility problems because of glyphosate, which is really common, that in 4 to 5 months of taking that, you'll pull the glyphosate out and you'll see human as well as animal fertility restored. So that becomes very important in that area. But the humic acid and the clinoptilolite are both very strong chelators and will pull it, but you need to be sure and take a good mineral supplement, because they're not, it's an indiscriminate removal of your micronutrients. So if you're taking those for any length of time, be sure and take a good micronutrient or good mineral supplement in that also. But it will pull the glyphosate. The only water treatment plant that will remove glyphosate that I'm aware of is the Personova one. They make one for residential use, fits under your counter, but reverse osmosis, none of those systems will remove glyphosate from water.
Hi. So I have a question for Dr. Porter and Dr. Huber about glyphosate and being now IARC in 2015 declared or said that glyphosate is now a probable carcinogen. And then California recently sort of upheld that in their own regulations. And I'm wondering if you feel that that might be what you think about that and how that may start to change the conversation that we're having around glyphosate.
Well, I certainly hope so. It's hard to say. People are scared of cancer. Unfortunately, they're not as scared enough as I would hope they would be about all the other consequences of this exposure. And so I don't have a generic answer other than should we keep on scaring people as much as we possibly can? There's plenty to do that, but we need people. I don't know whether people have to have something happen in their own family to make them change their mind. A lot of times that seems to be the only thing that will do it. And so it's a question of when are we going to reach that tipping point. I think we're getting very close to it.
I think the lawsuit, the non-Hodgkin's lymphoma lawsuits, we have 3 of those now, or the 37 have been consolidated. The first one that's gone, that's been filed is the California one. That's where we're getting a lot of this information. The early studies where Monsanto's have shown definitely that it's a carcinogen, certainly going to help. I think the second lawsuit will be end-stage kidney failure. I consulted in Guatemala and Brazil and some other countries. In El Salvador, 25% of their male workers will be, sugarcane workers will be dead this year from end-stage kidney failure. You look at our county, there, an agricultural county in Idaho, we have 5 satellite kidney dialysis clinics now where you didn't have any 20 years ago. Extremely, are becoming an extreme problem for us. I think it's the next range for class action lawsuit. I have some attorneys that estimate that the medical health fund that'll be required, trust fund for GMOs and glyphosate, when it finally comes together as it's starting to move, will exceed $1 trillion. The health impact in all these areas, from endocrine hormone disruption, you saw the one slide with the innards developing outside the body, typical glyphosate type of an injury. So common now that when doctors see the ultrasound, they know exactly what to do. You have to take that child with a cesarean, because it can't have a normal birth or it's all torn apart. But it's a standard procedure almost in many areas now, how to get those innards back in, usually about 3 surgeries. Sometimes they can get it in 2, but these are just common things that we're starting to take for granted. That's the problem. They 20 years ago, 30 years ago, extremely rare, that now doctors say, well, gee, we've always had these. We have standard operating procedures for moving to do it.
As a farmer, you get paid to produce pounds, not necessarily quality in our food. And we were at a pesticide meeting, and they're mainly chelators of minerals, pulling that out of our food supply. At what point do we get a possible quality standard on our food supply? And I know that organics is a step in the right direction, But if you look at their standards, they're procedural based, not results based. And how soon before they start looking at that? I think we have the labs and the technology to check it. And as a farmer, we're one of the few industries who you buy everything retail, you have to sell it wholesale, and you pay shipping both ways. Where if we had a quality standard and that was tied to your production goals and you were rewarded better, I think more farmers would definitely come on board. But I guess, do you know if there's any time that might be happening?
I think we have several things that are going to move that very rapidly. One is Joe Clapperton and Ken Hamilton's development of the Buechler system for rapid identification. You have a system right now to do the mineral analysis and vitamin analysis and everything. You have to have a $500,000 piece of equipment out there. With the Buechler edicts analysis now, you can take a leaf, or you can take mash up a potato, or you can take a soil sample or a single strand of hair, and you can tell what the mineral content is. And those instruments now are about $32,000. Jill's developing all the software for it, but it's going to make it so it's much more accessible. You'll get it into a lot of your large grocery chains would have the capability then of actually doing that type of nutrient analysis, that if you have to do all the extraction and everything else, becomes a prohibitory type of a process. I think that's the next step.
So we're supposed to have been at lunch 15 minutes ago. One more question. Do we have one more question? Burning question? Who gets it?
This is for Warren and Don, if you have some comments on this too. Can you talk a little bit more about autism rates and the prediction and the basic? Is it mostly the glyphosate, or is it everything?
Well, one of the things that happens when you're exposed to pesticides in general, and glyphosate possibly in particular, is the initiation of what we call oxidative stress. And that oxidative stress has recently been shown in the literature to start the degradation of the jelly, if you will, the microglia around nerves. That microglia begins to die, and as it dies, it produces its own toxins, which then alter the nerves themselves. The toxins get inside the nerves that it's surrounding. So you've got a positive feedback system that you've initiated with oxidative stress. And that could be behind at least part of the autism that we're seeing, a change in development, especially of the basal parts of the brain that are involved in filtering out stimuli.
I met with Dr. Dana Stanley at Queensland Central University last year, and meet with her again here in about 6 weeks. But she's been taking the gut microbiome and dissecting it out. With the molecular techniques now, we can start to identify a lot of those organisms that are there. She's completed her animal studies for autism and for type 1 diabetes. She has the organisms identified and run them through the rat studies, where they've been able, she gets complete cure of type 1 diabetes with just one organism or two organisms when she repopulates the gut with them. For autism, it's two organisms that complete absence of any of the symptoms in that area. She now has her proposal in to move that into the GI or into the gastrointestinal type areas with inflammatory bowel type diseases. She's completed the animal studies, but she hasn't identified all of the or she's identified the organism. She hasn't had the human trials on that like she has with the other. But with the autism, we've known that fecal transplants about 95% effective in curing 16 of the GI type related diseases such as autism and inflammatory bowel difficile diarrhea. The problem is that if you just do a general and fecal transplant, you can also have some really serious side effects if you get the wrong donor. So that you can get depression, you can get schizophrenic type effects if you get the wrong organisms in that general melee of things that are being added. But what Dana's done is she's identified the specific organisms. The problem is they're all very sensitive to glyphosate. And so you have to change your diet. And if you'll talk to the pediatricians, I have a son-in-law that's a pediatrician, says the first thing we do with autism is get them to change the diet, get them off, and in 3 to 4 days, you'll start seeing the improvement. He said some of them, it'll come very effective. With others, it's an improvement, but not a complete cure. But the GI tract, that 8th organ that we have, is really critical for the neurotransmitters, for all your aromatic amino acids. And Stephanie Seneff and Anthony Samsell and their 2 or 3 papers have connected all the biochemical dots in that area. Well, I can't see up there. Was there a question?